Okay, so. Okay, so hello, welcome everyone. I'm so very pleased you've joined us all for our lecture today. We're very lucky this month. We've had two webinars this month, and we'll be having a break in April though. So I'm Mila Stroganoff, and as programs director for the Friends of San Pedro Valley Park, I'm so very glad that you decided to join our ongoing natural history series. If you have missed any of our lectures, all is not lost. A great many of them are recorded and available if you go to our website, and that's friendsofsanpedrovalleypark.org. Other lectures will be added once our IT specialist returns. He's in India at the moment. As is our norm, we will address questions at the end of the lecture. So please do not hesitate to write your questions and place them in the Q&A section. And we will take as many as time permits. If possible, any comments or otherwise, please put those in chat so we don't get confused between the two. I'm so very, very delighted to have Dr. Joseph Szewczak with us. And he serves as a professor of biology in the Department of Biological Sciences at Cal Poly Humboldt in Arcata, California. Originally trained as an engineer, he has often called upon his technical fluency to solve problems relating to studying bats, birds, and other vertebrates. He has investigated bat physiology, distributions, and more recently their vocalizations, which motivated him to develop SonoBird and SonoBat software for analyzing bird and bat vocalizations. Dr. Joseph Szewczak is a prominent name in bat research for a variety of studies, but holds the greatest distinction as the creator of SonoBat. And even more recently, Joseph and his colleagues created a 3-year assessment of the status of the Townsend's big-eared bat for the California Department of Fish and Wildlife. In collaboration with Yosemite National Park, Joseph and his co-authors have completed a 3-year study to evaluate food webs and trophic levels in bats and birds along the Merced and Tuolumne Rivers. And so here we have Dr. Joseph Szewczak. Take it away. Thank you.
Okay. So I guess I get to share a screen then?
Yes.
Put this up here. Okay. So I've actually been out of doing Zoom lectures for the past year now. It's like I got used to doing this for a while, but so here we go, because I can't see any of you behind the screen. So I hope this is all going to work. All right, so yeah, so I just there's a lot of things that I could put into here, so I just brought a few things together which I think might be of interest, and I'll work through that. And when we get to the questions, maybe I can pull in some other things or take it from there. So so I'm calling it a little bit of bat pourri, if I may. So all right, so start off with what's with bats. So bats we put them into the group Chiroptera, which is Latin for hand wing, and we call them that because their wings are formed from their hands, and so they have all the original fingers here that we have. And if we were to look at the analogous bone structure between humans, bats, and birds, we can see that all the bones of our fingers are remaining in the bats. In birds, the bones have become reduced, and most of the wing is just dead tissue, which are scales. I mean, they're still attached, but it's not living tissue. The wings of the bats are living tissue, and as such, they as we can see in the previous view, we can see that there's vascularization in there, blood vessels running through there, and there's tiny little muscles that scrunch the skin up, the membrane up when it folds up. Joe, so the bats.
Joe, I'm sorry to interrupt, but we're not seeing the PowerPoint.
You're not?
No.
Well, that's a problem. It says I'm screen sharing. What are you seeing?
We're seeing just the full greens, the nature screen without the PowerPoint. Nothing has moved since the first screen.
Oh, all right. So, stop share. So let me try this because it's on a different, I've got a bigger screen. All right. Let's come back to here. Stop this. How about if we do it this way?
Okay, move it forward and see if we'll have a change of slide.
Yeah, let me, so I'm sharing now, and how about that?
Well, that's the original one we were seeing without any other ones.
How about that?
Still the same.
You're not getting anything.
No,
It's
You only,
the background screen is what
the
we're getting.
background screen is what we're seeing.
oh, okay. That's
And
what you're
nothing-
saying. All right,
and
I'm gonna
nothing
stop the
else.
share. Yeah, I think that it might be the problem because I was having two screens here.
Mhm. Now we see California Republic.
Yeah, all right, that's my stuff. Let's. I'm quitting the PowerPoint. I'm going to start that up again. That's not the one. So,
The California Republic is still up.
yeah.
Uh-huh. Do we have to do anything from here?
No, I don't think so.
No,
I think once you do
we're-
a share screen on your side, it's all in
Yeah.
your hands,
Yeah.
right?
Okay.
we're allowing Joe to share, so
Yes,
it's
yes.
up to him, I think.
Yeah. So you're still there. I'm still here. I'm going to share screen again.
Okay, so the
People can see my screen, and
yes. First slide,
you can see
the back screen.
all right, but the first side, you should be seeing a slide with my name on it.
No,
Well, why isn't,
Wait.
hold on. It looks like it's a permissions problem.
it's a which?
Permissions problem?
Yeah,
From whom? From me?
no, from my screen or something. It's not letting me do this.
Hmm. Well, it says at the top in the green that we are viewing your screen. Joe's.
Yeah, you're
that we're-
viewing the screen, but it's not letting you
It's not
see.
letting you move forward.
No, but you're seeing.
Uh,
Okay, what are you seeing now?
There's a white rectangle in the middle of things.
Yeah.
Let me check what's going on here.
Oh, wait. I think I see what's going on. All right, I'm gonna have to leave and come back in.
Okay, sounds good. Okay, so let's see, where is he?
Oh, it says he left.
Oh, he left, so I have to wait.
Yeah, there's only one panelist.
Yep. Yep. Okay, folks, just stay with us. We're just having some technical difficulty and this will be rectified very quickly.
Okay, back to two panelists. He's back.
Where is he though? I'm gonna find him.
Can you hear me again?
I can hear you. I need your name though, so I can, oh, there you are.
Okay, all right. So
right.
let me try this one more time.
Okay. Ah, things are beginning. I think it's a bat puri.
Bat Puli, all right. I am so sorry, everybody. My computer decided that it wanted me to give permissions to Zoom to show you something. All right, let's get back to where we were. All right, so we're starting off. Here's what I thought you were seeing. Here's a bat with his open wings, and it's Chiroptera hand wing, and we can see the blood vessels in the wing. And I was trying to make the point that this is all living tissue on a bat, unlike on a bird where we just have the feathers that have grown and are attached at their shafts, but not fully censored. So the bat's wing is fully censored, has fine little hairs over it, so it can feel the flow of the air over it. And all the fingers remain that we have in our hands. And so the bats can manipulate their wing just as we can manipulate our hands. And so this is what makes them able to do such maneuverable flight, because it's a fully instrumented and articulated flight surface that they have. It's like a aeronautic engineer's dream to be able to manipulate a wing in that way, as opposed to the way the birds do it. Now, bats belong to the mammalian order Chiroptera, as I mentioned, and in that there's over 1,400 species now described. And but to put that into perspective, that's 1 out of every 4 or 5 mammals on this planet is a bat. And we normally don't think of bats as being that numerous, but that's because we don't encounter them that often, because they're up in the dark where we can't see, or go into little spaces where we can't go. But an enormous part of importance to ecosystems around the world. But the sad story is that more than half the species are in moderate or severe decline. And while I've got this classification diagram up here, I want to point out that bats are distantly related to rodents. And so this notion of bats as being flying mice is quite apart, not a reality. It's just they're small little furry animals, and that's about as far as the association goes. Bats are much more advanced in terms of their brain structure, social structure, reproductive structure, skull structure, and so on. They're much more like us as primates, more closely related to us as primates than they are to mice. And so compared with mice, that's if you have a pair of mice and they have everything they need, at the end of the year, you're going to have many mice. But because they have litters, and the litters will have litters within a month or two. But if you have a pair of bats and you give them everything they need, at the end of the year, if everything goes well, you might have 3 bats, because they'll just have 1 birth a year. And it's typically just a single pup for most species. And they'll invest in that care for that pup throughout at least the 1 season. And so the low reproductive rate is something that is important in how we conserve and manage and look after bats on the environment, because we can't have something that happens to their population and expect them to recover. So our monitoring and conservation efforts have to be anticipatory. We have to know what's going on for them and what they need, and prevent a loss from happening before it happens. but they make up for their low reproductive rate with longevity. So a small little 7 to 9 gram bat can live for 30 to 40 years. The current record in the US it's now actually exceeded 34 years. And I think in Europe, it's now up to about 43 years. And this is in spite of the fact that we do not band or mark bats that much. These are still from very small sample of records that we get this longevity. And so bats, there's a lot of research going on. My colleague, Manuel Vasquez at UC Berkeley has been doing work into the molecular and genetic mechanisms that enable this longevity, which is impressive for an animal their size. So, but why should we care about bats? Because they're such wonderful creatures, as this one here I'm holding in England can show you. But what is it really that matters to us? Well, they play essential roles in ecosystems throughout the world and in those high numbers. And for example, pollination. And for some people, it's just enough to know, no hay murciélagos, no hay tequila. And that's enough to convince them to like bats. And in fact, if you look well enough now, you can find bat-friendly tequila where the crops, the people that operate the crops are leaving a certain number of the agave to actually come to flower to support the bats, and in turn that supports diversity in the agave flowers. But so many of our important tropical vegetables are pollinated by bats or the seed dispersals. But if we look at one, here's one of our nectar feeding bats. I'm holding it here in Arizona, and we had just captured it and we were processing it, about to let it go. And I thought, well, let's give it a little drink before it takes off. And it's just like this wild animal here is just totally calm in our hands and just drinking up the little bit of nectar offering that we're given to them here. But the bats that pollinate, so most of the bats that are involved in pollination and fruit eating are in the tropical parts of the world, but all of the dominant rainforests in the tropical parts of our planet are dependent upon bats for either pollination or for seed dispersal. And so they're a major part and importance in maintaining our tropical rainforests, which are vital to the overall like carbon cycling and overall climate, for that respect, of our planet. So yes, there are bats that drink blood, as you can see these ones here, enjoying my toes. And yes, that's the aftermath, and that is me there. This is something I did for a show on vampire bats for the Discovery Channel. But out of those 1,400 bats, there's only 3 species that have adapted to drinking blood, and those are very specialized adaptations. And the bigger question is, why is it only 3 species and only in the New World here? And that's the more interesting ecological question. But it is the seed dispersal and pollination in the tropical parts of the world that are, a really big importance of what they do in these parts of the world. And of course, in the tropics, that are much more numerous in terms of species than they are in the temperate parts of the world. But they are throughout the temperate parts of the world. And in the temperate parts of the world, they're not so involved in pollination or seed and fruit eating, but insect prey, predate upon insects, and some of the most important insect controllers that we have. And often not known by many people is the fact that the aerial density of insects is so much greater at night than it is during the day. And that's because it's very dangerous for a very small bodied animal like an insect to be out in direct sunlight. The surface to volume ratio means that they'll lose moisture and dry out very quickly. And so most insects don't take flight until night. And bats, having developed their ability to echolocate and detect objects in the dark, can prey upon those insects. And they go to town on the insects at night and have tapped into that resource. And about a decade ago, some colleagues evaluated this and looked at it in terms of, the economic benefit, and the bats benefits, agriculture by billions of dollars. And this was 10 years ago. And today's dollars, this is more on the order of about $40 billion worth of benefit and reduction of agricultural pests and increased productivity. in agriculture. And that's just talking about agriculture. It's not even talking about the benefits that the bats do for forest health and eliminating many of the moths, caterpillars, and so on that damage forest trees. So that in mind, we've been actually working just recently with some of our state senators here and working on getting this bat here, Pound bats, to be recognized as our state bat. We do not have a state bat, and we are in progress of trying to get a state bat. And some of you may come across some of this, and I hope that you can support this initiative. Pound bats are really awesome, charismatic bats. They're the golden bats, so we think it's appropriate for the Golden State. And these bats are leaners. They actually come down to the ground and will take prey off the ground, such as scorpions, as this one is showing here in the mouth, and centipedes and things like that. So, but when we go out and study bats, it's not easy. So even here in this image, there's 9 bats on that tree, and these particular bats down in the tropics are actually spend their day roosting just out in the open like this. And unless you really can key in on this to see them, you're apt to miss them. So this is one of the most difficult taxa to study as a biologist, because after all, you go out at night and this is what you see for the bats, or don't see, because they're out of sight. They're in the dark where we can't see. They're out of reach up in the airspace where we don't go. And they're mostly silent, making sounds that we can't hear. So just about everything that we do to try to study and understand these animals, we have to adapt some kind of technology to expand our own corporeal sensory systems to be able to investigate what they're doing. And so how do we do that? So here we have one of our bats here, the hoary bat. It's all over North America, handsome bat. These bats roost up in the foliage at the tops of trees, and another place where we don't go. So they're very difficult to track and monitor. So one of the really important resources for us to help preserve them is to know where they roost, because they are very vulnerable when they're roosting. And most people assume that that's going to be in caves, but actually caves are more of the minority of bats, places where bats roost. But it is one of the places where humans and bats go and end up in the same place and can see each other. But it's actually not the most common place where they roost. For example, we've got a dozen bats up here, Northern California, Redwood Coast, and we don't have caves, in any numerous fashion up here at all. So where are they going? Well, crevice roosting is perhaps the most common place where they go. And so anytime you see any exposed rock and you see some fractures or joints, crevices in the rocks, those are the kind of places where bats are often roosting. And the deeper the crack, the more they can go into it and move back and forth to get just the right temperature profile they like. And not just rocks, but trees. Trees, snags, when they get to certain stages of decay, the bark will loosen, and underneath spalling bark like that, there could be a couple hundred bats roosting up in there, and they will move about the forest, finding different roost locations. And of course, this crevice roosting habit has been adopted by many of our bats with our structures, buildings as well, and in particular bridges. Bridges often spanning really good foraging areas for the bats. This is looking up into the crevice structure on one of our bridges, and these are free-tailed bats that are quite numerous in our state, and in many places these are the ones that we can see great exit flights, as right here at the Yolo Causeway between Davis and California. We've been trying to get a good count on this. Layla Harris at UC Davis has been organizing some surveys of just counting underneath the bridge in here, and there's estimates there's maybe 250,000 or more bats on this bridge structure. And it's a great thing to go out there anytime you're up there. The western end on the Davis side is a good place to go almost any summer night. You get there in the evening and you can watch the bats come out. They'll come all the way down. They're roosting all along the length of that structure, but they'll all fly under the bridge and come out en masse together like this and just form these rivers flying off into the sky. And they'll go off and fly maybe as many as 60 to 100 miles in the night, and just all over the Central Valley and foraging over places like the rice paddies and so on. It's a great sight to see wildlife in abundance. So how do we go about this? So capture is one way of doing this, and then various imaging technologies, as well as listening. And so listening, we've been doing a lot of work on this in recent years, is understanding what it is that we can do to actually listen to them and track them. We've got thermal imaging equipment like this now, where we can actually watch bats coming out of the roost that would otherwise be difficult to see. And anytime you get to a roost like this and trying to watch the bats come out, the bats will come out right about the time when it gets too dark to see them. And so it's nice to have something else that can help us do it. But very often we don't know where the roosts are, and the way that we find our way to the roost, and we've learned so much about where the different places that bats roost, is being able to capture them when they're out and about foraging, put tiny little, like 1 gram or less radio transmitters on them, and then be able to track them back and find their roost. But to do this and to do a lot of other work, we have to capture them, as we're doing here. And I should mention, anytime you see these pictures where we're not using gloves on them, these are kind of legacy pictures from years ago, because currently, over the last decade or so, we've gotten better at protecting them from pathogens and back and forth. And in particular, that happened when we started having the white nose fungus that we want to prevent passing along. So typically we use mist nets, sometimes like here, in closed environments, I can just catch them a little easier like this, as I'm doing for some of my students. That comes from working with them long enough that you kind of know what they're going to be doing. All right, but and our mist nets can get quite large. We have these sets where we stack 3 nets up on the top of each other here, as we're doing here. This is a site out in Eastern Washington. But mist netting lets us catch them, but it's not any, it doesn't really help us at all for doing any sort of long-term monitoring, because once you've captured the bat, they're going to remember that there was an obstacle there, and they're going to be, basically won't be able to catch them again. So you can't continue doing this repeatedly throughout the season to monitor their activity. And so that's where these other things come in. And so curiously, despite having a lot of species that have structures like these ears here, it remained a mystery for a very long time to understand how they're getting around in the dark. And it really, this is something we all know now. We teach school children this, is that bats are making sounds and they're listening to the echolocation. And we think of this as being like one of those like enormously, wonderful things that some animals do, that, special abilities that animals have that we don't have. And you all could learn to echolocate yourself. And a lot of humans, particularly some blind humans, have learned how to do this quite handily. And if you make a clicking sound, close your eyes and pass your hand in front of your face, you will know when it's there or not. And a lot of people have promoted, we should all learn how to do this, because it would help us find our way out of a burning building where lights wouldn't help us. But just to move on, what does this do for the bats? Well, what this does for the bats is that it enables them to roost in places where vision-oriented predators can't get them, because when bats are roosting, their wings aren't, useful for defending themselves, and they'll go into a physiological torpor where it takes them a while to respond. So that helps them. But the biggest thing is that it opens up this enormous feeding niche of being able to go after that huge prey cache that's available in the nighttime sky. And that's quite good. It enables them to do this. But the problem with that, of course, is that then your prey can sometimes hear you coming. And so a lot of moths, for example, about half the moths of the world can recognize when bats are coming, and will actually then avoid that. They'll hear the bats coming, and they will it'll inhibit their wing muscles, which make them a little flutter out of the air. And you can try that sometime when you see a lot of moths flying around a light or something. Take out a set of keys, and when you shake those keys, you can actually generate sufficient ultrasound to cause that mechanism in moths to happen. And you'll see a number of these moths just sort of flutter out of the air, and that's their bat avoidance procedure or reflex, and that it works well enough that it's been a conserved trait, and they pass that along. But what is good for us is that we can listen to them. And bats generate their sound in their vocal cords, just as we do. But they have two ways of pushing it out. So there are oral emitters, and then there are nasal emitters. That is a more sophisticated pattern of doing this, that by projecting it out the nose, then they can still be using their mouth or chewing up the thing they just caught, for example. And the bats that you see that have like the very elaborate nose leaves, those are the nasal emitters, and they're actually helping to project and focus the sound that they're putting out. And but I want you to I want to express understanding that when bats are using echolocation, though, that they're using it in the way very similar to what we do with our headlight or our I should say our little spotlight when we're out at night. And I'll make you another note of that. But what we do is this is showing a sequence of bat echolocation calls, and it's changing it up as it goes through the sequence here. And I should say that these calls there's a lot more time between the calls and shown as representation. When we look at them and view them, we sort of scrunch them all together and take the empty space between them so that we can see the progression of these sequences. Because what they're actually doing is shown in this. This is a real-time representation, so we can't see the shape of the calls as well, but we can see the progression. And when they're just out flying along, putting out a sequence of calls, this is what we call, refer to as their search phase. They're like putting out calls, listening for any echoes to come back. When they hear an echo, then they will start upping the repetition rate of the calls that they're making. That's what we call the approach phase. So they're steering toward the object, and then they're assessing the object at this point. And when they do so, then they're actually determining, like, is that the kind of prey item they would like or not? Because they're actually very selective in the prey they take. If they decide they want to take it, and their echolocation is so well, it gives them such resolution that during that approach phase, they're not just like tracking the insect, they're getting close enough to it with their echolocation, actually able to image the insect and recognize what kind it is. And very often they'll then decide, oh, I don't like that kind. They'll go on. But if they decide they like it, then they'll go into this final little sequence we call the terminal phase, which is this very rapid sequence of pulses, which helps them to track and catch the insect. And so they're using their echolocation just like you would flashlight. If instead of a continuous beam, you could just like flash it. And when you're walking along fine, you're okay, sort of flash, flash, flash. But if I throw a Frisbee to you, you're going to go flash, 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 flash to track it. And that's what they're doing with their sounds here. And so we can play this. I think this will work. I go. Not sure if you're able to hear that or not, but that's the whole sequence, what's going on, search phase through terminal phase. And I want to point out now that you can hear that because we've slowed it down so that the frequencies that we normally can't hear, they're too high for us to hear, are now slowed down, and that means we can hear them, but it's slowed down by a factor of 10. So this whole sequence of events we just looked at here all happened in less than 1 second. In fact, 3/4 of a second. And in real time, it's like this. And then we hear that like buzz at the end there. Okay, this is watching little video here. One of my colleagues here, we can see this in action. And this is a red bat, and you see it's catching it with that membrane between its hind legs. That's red bats are that. Most bats, though, will use the wing like a catcher's mitt, where they'll get close to it and they'll swing out their wing and sort of grab the insect and then sort of push it down into that membrane between their legs, and then they'll grab it and eat it from there. And it turns out that about 95% of bats are right-winged, and then about 5% are lefties, just like us. All right, so when we started having the tools and technology to listen to them, then we got all excited because we've been doing this for history with birds, listening to birds and then being able to recognize them. But it turns out to be a much more difficult task with bats. So because birds are using their calls and their songs as identifiers, which is to say they're selected to say, this is who I am, this is where I am. There's no constraint on complexity of the sounds. But bats in their hand are using their calls for this specific task of information acquisition. And to best solve that, it's going to be a single little chirp. So there's no overlapping segments because that messes up on the timing of what's coming back, which gives them the assessment of the distance to the echo producing object. And so many of our species have converged on having very similar call pulses because that's the best solution for the problem, so that there's no selective pressure whatsoever for them to be different than a number of species. The selective pressure is to solve this problem of getting information back from these echoes. And so this has made it quite difficult for us to be able to tell them apart. And so we have to look at, little nuances in these calls and that they're changing the calls according to what they're doing to add another level of complexity to this. And just to fill in, you might have heard this from people blind as a bat, and we should put that aside. That's only accurate insofar as if you're referring to a particular piece of sporting equipment that's used in the spring. But if we can say that, then bats can go around saying deaf as a human. And I bring this up just because bats have this ability to be able to echolocate in the dark. By no means leaves them as not having an ability to see. And in fact, bats see much better than we do in the dark. And they don't have the color vision because they just mostly have the more light sensitive rods in their retina. But they combine these sensory, all biological systems are going to use as much sensory information as available to them for best success. And bats use, as I mentioned, their echolocation like you do your light at night. It works very well to give you detailed imaging of what's up close to you, but to get your landscape scale perception, your light is not going to shine across that meadow and illuminate those trees, but you're still aware of those trees because of the shadow and against the night sky. You know where you are. And so bats are using their visual perception as clues to where they are in the landscape. And these bats that are going out foraging miles and miles away from their roost, they're not going to find their way back with echolocation, which is only going to be able to, give them, 10 to 30 feet around them to be able to image things. So you use your light to avoid tripping over things on the ground right in front of you, but it's not going to find your way home. Just using your light give you an idea of what the perception the bats have for their echolocation would be. Imagine now using your light on a very foggy night where your light just doesn't go that far. And that's sort of the way the bats have it with their echolocation. So they're mostly using their visual clues, but they can detect the little insect objects and other obstacles going into a dark space like a forest using their echolocation. So they'll change up their calls according to what they're doing. We've got bats flying up high in the air versus bats flying in around a clutter in a forest or other cluttered environment. And there's different shapes to their calls, which best fit those different tasks. And that helps us to interpret some of their ecology of what they're doing. But again, to put it into perspective, here's a number of our California species here on a scale, a sonogram scale, where we're looking at the time on the x-axis and frequency on the y-axis, vertical. And down here at 20 kilohertz, that's the upper range of human hearing. So everything that we're hearing is below that level. And this putting the bats in perspective. And we have one bat here, the spotted bat, that and a couple of other species that we have. California do actually make calls that we can hear with our human ears, but that's more the exception than the rule. But down at the bottom here in the red, that's a sonogram of me saying the word bat. So in the time it takes me to say bat, all those animals are able to make their calls much quicker in that time sense. So everything they're doing is operating this much higher speed way of life than what we have. Incredible little processing brains that they have. Okay, so we've got bats. When they're the ones that forage up in the open air, we use these lower frequencies. They travel farther in the air, give them more perception. And the ones operating in this cluttered environment sweep down through frequencies much more. And that gives them a more detailed interpretation from their echoes of what's going on around them. And for us to identify them, it's much more difficult because they're changing up as they do. So just as the open air foraging bats are doing different things than the bats flying in the clutter, even within a species, they will adjust their calls somewhat along that range. And so these are all call samples taken from the same species, but different individuals operating in different locations, different situations. And so our task at recognizing bats is to be able to recognize all these different call types that belong to a certain species, or what we call the repertoire call types for that species. Some of them will overlap with call types of other species within the other species repertoire, and so on. So we have to pick out which ones are actually species discriminating to make our basis for what kind of a bat we have and interpret in what they're doing. So that's what makes it difficult for us. And it turns out it's actually rather easy to record the bats because they're making very loud sounds. In fact, if we could hear the bats, people would probably have persecuted bats even worse over the course of history, because there would just be this total racket of sound that's going on at night sky, and we've just been blissfully unaware of it. So we can tune into it and we record it, but it's very difficult to calibrate our recognition systems with these animals, because it's easy to record, but it's very difficult to know who said it. And so over the last, starting in actually in the 1990s, I started recording them and tracking them and trying to work out who is saying it to build up our library of reference recordings so we know who's saying what. And so the obvious thing is that you catch the bat and you let it go and record it. Well, it turns out when they're just taking off, initiating flight, they're not making the kind of calls they make when they're just flying overhead in normal operating airspace. And you think the bat is going to fly off to the right here, but in fact, the bat has another idea. It flies off to the left, and you got your microphone over there on the right. So you got to have lots of microphones around to try to get it. But still, when they're just taking off, they're disoriented. They're making more calls that are like that approach phase rather than their open air search phase, which is what we record when we go out and just passively record for them. So what do? Well, we can fly them. I developed this little way of flying them on this mini bungee that's an elastic sewing thread. Or sometimes we can spotlight them and recognize them by their shape and call patterns or flight patterns. But the best way we've done this is to actually catch the bats, put little light tags on them, and then release them. And then hopefully they'll stay around long enough, settle into normal flight, that we can then track them, record them, and have a number of recording sites around to do this. And so to do this, we catch the bats. Here's a couple of my graduate students putting up some nets in Arkansas some years back. And then we make these little LED light tags, apply them with some goopy glue that's non-toxic, and we put it on their chest because it's just temporary. And when they get back off flight, they can just remove it themselves. And we've seen them do that. And then we have these mobile recording units that we set out in a number of groups around an open area. And you have to track the bat, see its movements, and put it together with the sound you're recording. Because just because you can see it doesn't mean that's the one you're recording, because there could be a louder bat closer to your microphone than the one you're looking at. So you got to put the two together to be able to confirm that's the animal you have. And so my colleague John Changer has made some of these nice time exposure pictures of showing me releasing the bat, and then we've got people recording the bats at different locations. This is one here, and you can see some of the recorders in the background. And this is up north in the Modoc Forest. Bat took off, oriented itself, then took off, flew through the trees, back out fir trees and right over one of the recorders there. Okay, so long-term recording, we go out, we can set up our microphones in places where we got bats flying around. We can get the ones that are doing the edge foraging, the open air foraging, and flying around, get some open sky to get the ones flying over in the open airspace. And that gives us a good sampling of what's going on around us. And so we'll set up our microphones in places where we can avoid echoes from the surface objects. And from this, we can get a lot of info as to what's going on with the bats and put it together. And we can actually even set these units up and do what we call mobile transects, where we go out and we drive a pattern at a certain routes and certain speed. And then we can put the data together as we're done here and show what bats we see at different locations on the landscape. So that's some of what we can do for the monitoring and just by listening to them. And just briefly jump into a couple other things that we can do. And we working with Sarah Stock and Breezy Jackson at Yosemite initiated this Big Wall Bats program, where we've got the climbing rangers and docents trained now to like be aware of some of the bats. We're starting to work out where we have these little probes that we can do. And the first time we went up and worked with them training, we're teaching them to use these little video probes that can go in the crevices to check for the bats. And we went to a site just barely off the valley floor there just to train this. And we're up there, we're looking. And sure enough, right where we got started, first time out, there's a pallid bat right there looking at us. And we stayed at that site in the evening and watched about two or three dozen of them come out from that site. And so now we've been able to track them, establish, and starting to look at the ecology of how much they're using on these vertical surfaces. And more recently, still not knowing where a lot of our Western bats go in the winter, being able to attach radios to some of them late season and being able to track them up to some of these sites on the cliffs to be able to find where they're heading out and staying for the winter. And that's again helping us to understand where they're going. And we mentioned the food web study, and this was the food webs are, of course, we've got what we call primary production. Primary producers, which are the plants, they're deriving the energy from the sun, and something eats the plant and something might eat the something that ate the plant and so on. So we talk about, the primary consumers, the secondary consumers, tertiary and so on. And these are the food webs. And the trophic level is each time something eats something else, that's going up a trophic level. And so the farther you get up in trophic levels, the more energy it takes to get to that level, because it's multiplied from everything that's below. And so this was something we investigated in the two river systems in Yosemite. And it was an interesting project, because this was actually a project in which we were asked to use science to help direct public policy. And the public policy is, what kind of a flood release do you do from the Hetch Hetchy Dam? Because the Tuolumne is a regulated river that has a dam on it. The Merced is a natural river, it's unregulated. And so the question here was, when you've got a dam, flooding events are natural events that occur on rivers, and they help to determine what kind of invertebrate communities you have, and that affects the plants, the animals, and the things that eat them. And so the question here was what is the best flood release regime to do out of the dam? Is it like a long sustained flood, or is it like a big gully washing type of flood? And we were able to establish and go down and do the field studies in there and set up and catch not just bats, but birds. And we're looking at able to use stable isotopes of oxygen, carbon, and hydrogen to be able to determine the trophic levels, as well as what is the primary production source. So you can actually track from those isotopes whether the basis of their food web was from terrestrial vegetation or from aquatic vegetation. And we're actually able to determine that some of the bats are actually more dependent upon the aquatic vegetation than some of the trout that are in the river, which was kind of an amazing result. And we did that by sampling, basically creating like field laboratories to take some small blood samples from the bats and from the birds. And it just only took like 10 microliters of blood to do this. And then we could release them and take care of them. And we were able to track and find that the trophic levels of the bats varied anywhere from like 3 to all the way up 6 trophic positions, which is quite remarkable to try to figure out who ate what to come up all the way up to there. And a lot of people were involved in that study. And so I just want to like note that this was just not me doing this. This is a lot of people involved in doing this. And I think, we're about filled out at the hour, and I want to leave some room for questions. So I could keep going on because I love this stuff, but I want to like keep it open for everybody else.
Right. So we will head to the questions. You'll have to excuse me. I have a bit of a raspy throat today. Before we get into the questions, though, you were mentioning the fact that bats can stop breathing for 2.5 hours.
Oh, yeah, that was some of my earlier studies doing the physiology of torpor. And yeah, so I elucidated a method that when the bats are in torpor and they bring their body temperature down to very cold temperatures, reduces their metabolism, that they have a mechanism that they can basically passively take in the oxygen they need down their airway by diffusion. And it's actually a similar mechanism to how insects breathe with their little air channels that they have in their bodies. And it's just a small little oxygen gets in that way, but when they're in that very low metabolic demand, it's sufficient to meet their oxygen needs. But they eventually have to breathe because that mechanism does not work as well for getting the CO2 or carbon dioxide out. So they still eventually have to breathe to exhaust the CO2. But I was able to document that they can go as long as 2.5 hours without taking a mechanical ventilation. And so just to note that anytime you see a bat that's hibernating in torpor, that you may have to look a very long time to see it breathe. And so just because you don't see it breathe does not mean it's dead.
So is torpor when they're sleeping as well? Or is
well, no, torpor is how they endure like a long, the winter.
the hibernation?
Yeah, it's hibernation,
Or
but torpor and
estivation.
hibernation are actually a little
Estivation.
different. Yeah. Like, I'm sorry.
Estivation.
Estivation is the way animals make it through a hot, dry season. Hibernation is making it through a cold season, a winter.
Okay, so let's see what we have in questions. Okay, so the first one had to deal. Brief evolutionary history. What ancestors like? When split off? This is
It goes all the way back, and they branch off about the time of in the mammalian history of tree shrews. And so bats probably came about from tree shrew, little tree shrews, small little mammals that were climbing up and down like the trees, and were probably jumping out at insect prey and started getting fingers that were long enough to grab the prey. And ultimately, then they were able to develop a little bit of gliding ability from that, and then maneuver a little bit as they went to get the insects. And that just kept selecting and improving upon it. And pretty soon, they developed out of that mode powered flight because they kept getting better at that. And then once they got to the point of powered flight, boom, they took off. And so it's a difficult thing to track in the fossil record, because once they got to that point, it was such a successful strategy, and being able to fly, they could radiate out across the planet. And, it's just like in a blink in the fossil record, and then they're everywhere. and getting into expanding into all kinds of different environmental niches and activities.
So there was no coevolutionary lines. There was just one line that went in.
It now looks like one line. For a while, it looked the two main branches, the megachiropter and the microchiropter. The megachiropter, like the large fruit bats, flying foxes you see in Eurasia. And back in the 80s, there was thinking that they were maybe had independently evolved. And modern molecular genetics has shown that no, that everything comes out of a single origin.
Oh, well, that's good to know. Okay, that's all right. So William is also interested about bat house design and positioning.
Yeah. So bats, unlike birds that when they fledge, they go off and set up their own housekeeping. Bats don't do that. Most of our bats are communal. And so they will just join and stay within the colony. And so, but bat houses are sort of like giving them housing insurance. So just putting up a bat house doesn't necessarily mean they'll always move into there because the bats aren't going around looking for new spots. But if it's a really good ideal location, has the right conditions, and it's conditions that matter more than just the space, they will occupy it. And the conditions they like the most are warm. I mean, warm, not to the point of too hot, but like warmer than ambient temperature usually. And that's like up to about body temperature, closer to body temperature. Mid 90 Fahrenheit is like their thermal neutral temperature, where they're neither too hot nor too cold. And so you don't want it to get too hot in the daytime. You don't want it to get too cold at night. And so that's why very large structures like attics and bridges and so on are preferred, because their temperature, buffering the temperature changes throughout a day. And so often just a smaller house attached to a larger structure like a building will help give it some thermal buffering. But just putting bat houses up on poles and so on, you can't get that kind of temperature regime that's good. But anyone that's interested in it, I would say go to the website for Bat Conservation International, batcon.org, BATCON.org, and they have a lot of resources available. And as does Bat Management and Conservation, another website.
I've heard of bat houses and not too much success with them, unfortunately.
Yeah, well, it's kind of thing you can have. You can put up a bat house and it may be unoccupied for years, and then suddenly it's occupied. And what's probably happened is they had a preferred roost somewhere, and they got evicted from that roost, or they lost that roost. But if you've got a bat house up, the bats are going to come around. They're going to know it. Remember, these animals are living decades, and so they know what's going on in their area. And they're able to communicate it to each other. And so if you put up a bat house, it's going to get checked out and they're going to know it's there. And so I like to think of bat houses as giving them housing insurance, because you're providing a safe refuge for them in case something happens to where their preferred location, or you may just hit on having a really good combination and they like it there, and then they might move in.
So Vernon is asking about, well, he's mentioning the fact that he took a trip down the Paria River in Arizona, and they were awakened at 4 a.m. one morning by the sound of bats circling the cliffs over their heads. And he says, I had not expected to hear them. How many of them?
Those are probably down there. Those are likely to be spotted bats. Or Allen's lappet-eared bats. There's two species down there that talk that, and also our massive bats are also sound. So if it's sort of a slower cadence, like tweet, 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 then it's a massive bat. But it's kind of a tick, 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 tick sound. Those are like a spotted bat or a lappet-eared bat. Both spotted bats and massive bats you can hear in Yosemite Valley in the summer. And so that's a great place to go if you just want to hear bats.
All right. Well, Yvonne is asking where we will hear this recording. The recording will be up on the Friends website, the Friends of San Pedro Valley Park.org. As soon as our IT specialist comes back from his trip. And so that may be in mid-April, end of April. But yes, it will be up. Do both parents, Joanne is asking, do both parents care for pups?
It varies by species, but for most of our bats, the female, the communal species, will form what we call maternity colonies, which are almost exclusively females. The females will share in rearing the bats, and like when they go out to forage, they'll put the pups all together in a little nursery group, and there will always be one female tending that nursery group, and she won't leave until she gets relieved by another female. But there are some species, and there's one notable species, I can't remember the name of it, but the males actually lactate and contribute to rearing the young.
Well, that's original. Okay.
Well, yeah, it's rare for that to happen, but it's a funny thing that like all male mammals have the equipment, and it just needs a
trigger.
biochemical trigger to make it happen.
Yeah, exactly. And do bats carry rabies?
Yeah, all wild mammals carry rabies, and bats have a much worse reputation for it than is deserved. But and all of us, anyone doing mammalogy work or bat work, it's just simple to get, prophylactic vaccination for rabies. And now it used to be 3, now it's just 2 shots, like getting two tetanus shots. So it's like the one surely deadly disease in this planet that you can get a simple vaccination for, and always, curious that we don't have more people that do that. But bats actually have a lower incidence of rabies than do foxes and raccoons and skunks and so on. But yeah, but it's a situation. I like to tell people, think of it as like bats. It's like a bare electric wire. You can't tell by looking at it if it can harm you, but it's not going to harm you if you don't touch it. So that's just a thing. But the sad part about bats and rabies is that most bats, at the end stage of rabies, animals either go into a ferocious stage where they get all gnarly, Cujo the dog kind of thing, or what's called dumb rabies, where they just become weak and listless. And most bats go into that dumb stage where people will find bats down on the ground because they become too weakened to fly. And like, here's this little passive animal, and they want to help it. But when you grab it, that's when the bat may respond reflexively, as threatened to bite. And so anytime you find a downed animal, call your wildlife care rehabber people, but don't handle it with bare hands. Use something, get it into a box, or use some, very, strong gloves to prevent that. But it's getting rabies is, like HIV or something. It requires like a body fluid to body fluid transmission. And so long as you don't handle the animals directly, you're fine. Well,
you folks who study bats and are handling them even with gloves, do you have to have yearly injections?
Not yearly injections, but what we do is like on a regular schedule every other year or so, we get our titer checked so that we know we have an active antibody level. Yeah, I was vaccinated in 1984. I've never had a clinical booster. My titer has remained high.
Oh, that's good. Yeah, okay. That's all right. And then what about the white nose virus? It's
not a virus, it's a fungus.
I'm sorry, fungus. Okay.
Yeah, white nose syndrome. It's a fungus. It was first introduced onto this continent in upstate New York. It has now progressed all the way across the continent. And the insidiousness about that is that it's a cold-loving fungus. So it can't thrive, it can't survive above 20 degrees Celsius, which is about 50 some degrees. And so it does best close to the freezing point. And so it only affects bats that are hibernating. So when bats go into hibernation and they reduce their body temperature down to low temperatures and they're during torpor, that's when the fungus gets on their tissues and it progresses to the point where it irritates them, and that causes them to arouse from torpor, have to expend the energy to bring their body temperature up. and they go through too many cycles of that during the winter, and they basically starve to death. So like most funguses, it doesn't directly kill, but it's a secondary effect that kills. And that is why we have had this initiative, like I mentioned, in Yosemite, for trying to track them to where they're going in the winter, because we need to find out more about their wintering ecology to be able to help track them. So where it first happened in the northeastern North America, and where it's been most affected, and just like, you know, the most common species has gone to emergency listing, is because the bats would hibernate in like huge masses in these caverns, you know, tens of thousands of them together. And any kind of disease agent, when you put it in close proximity, it can spread. And so it just like quickly affected the eastern species. Out west here, our bats we don't know any area where our bats hibernate together in large groups like that. They're more scattered in isolated rock crevices here and there. And that's probably going to help them to be less affected by this, but we still don't know. It's only been some recent years since it got here. It's like 2016, we had our first record of it in California. One of my grad students has been tracking those bats from that roost where we first recognized it, and trying to like understand where they are going in the winter. And
transmission is from bat to bat, from
bat to bat and location to location. So it's the fungus is related to a group of soil fungi. And so when the bats come out and warm up in the summer, they completely shed themselves of the fungus and its spores, because they're not viable at high temperatures. But when they return to these spaces where they hibernate, they get reinfected because the fungus persists in these colder underground locations.
Let's go and see what other things we have here. And Vernon is asking where we can find your taxonomy evolution chart that you showed during your lecture.
There's a number of publications where you can find that if you just search natural history, bats, mammalian let's see, the genetics and then the evolutionary history of mammals. You can come across those things.
Let's see. Let's see. Any bats protected by CEQA, and how is it?
Bats are all protected, not explicitly protected under CEQA, but they count as species that need to be considered for CEQA. Actually, existing right now, the best, one of the best legislative protections for bats actually has to do with stream alterations, because of their relationship to streams and dependency upon streams and foraging on streams and so on. That has in some cases helped, but no, we do not have any listed bats in California. I can tell you a lot of the biologists that work with bats believe we should have them, and many qualify for them, but they've been disregarded because we don't have the same historical records for bats, because they've been historically so difficult to study. And if you look at what bats are federally listed, they're all cave roosting bats, because we have records of people going in the caves 100 years ago and could see how many bats there were and make records of that. And then you look at today and can see it's changed. But like we've got a hoary bat and red bat in California that probably should be listed, but we don't have records of them from hundreds of years ago. For red bats, there's like some historic accounts of them being in clouds of flocks, like flying up and down the river corridors of the Central Valley before it was all changed, in the 1800s. No one living has ever witnessed those kinds of numbers. And these red bats are dependent upon the cottonwood galleries that used to line all the river in corridors of California. And there's like only 2 or 3% remaining cottonwood galleries in the state. and probably similar numbers for the bats compared to how it naturally was.
And in the Bay Area and Pacifica, in this area, do we have a certain number of species of bats that are?
Well, there's 25, 26 bat species in California, depending upon how you count them and which ones just enter in. And right around where you are, there's probably about 14 species.
14?
Yeah.
Gee whittakers. Okay. And there's a huge decline in insects.
Yeah.
In the world.
Yeah.
How is this affecting bats and their consumption of insects?
We've been seeing bat numbers go down over the years, and that's again a big part of it.
Uh-huh.
Yeah.
And you were mentioning aquatic vegetation as being the preeminent source for food for the.
Well, not all the species, but for a lot of the species that, it's like when we want to like study bats, we often go to where the water is, because where the water is, you have the most, plant production. You have the most plant production, you have the most insects, and the bats get from there. And so it's just following the food web and to its source.
Mm. And did the SonoBat, in order to create this, the piece of equipment that you did it take you long to create?
I started puttering with that in the early 1990s, and realized that was the approach to do this. But back in the day, with 16 megahertz processors, to generate one of those sonograms would take 5 minutes, and so it was wholly impractical. But I realized that I thought, well, computers should be getting better someday, and I kept recording that kind of data, knowing that ultimately we'll probably be able to use it. And in the early 2000s, computer technology had progressed to the point where that's when I really started working on the program further, and have developed it along to where it is now.
One had to catch up with the other.
Yeah.
Okay. Let me just and with birds and this whole idea of going out in the springtime. I mean, I've read and I've studied about the fact that it disrupts when you want to have a response from them while they're mating and having their, building nests and so on and so forth. How disruptive is it actually to have human beings going out into open spaces and beckoning the birds or trying to arouse them to respond?
Oh, like doing playback calls for the birds?
Yes.
Yeah. Well, that's not recommended.
No.
Yeah, because it disrupts them. Surely. And it's, it's like many things, it's a dosage response. So a little bit of it is probably not much of an effect, but you keep doing that repeatedly. And yeah, I mean, that can happen when we have rare bird sightings. Everybody goes out there and they want to see the rare bird, and they basically harass it to be able to see it. So, we have to, hold ourselves back sometimes.
Well, let me just, I think we've got some more here. Have you still got time?
Yeah.
Yeah. Okay, good.
It's Saturday.
It's are there serious predators? William is asking, are there serious predators to bats since they fly so fast?
There, other than a couple of bats in the tropics that regularly prey upon bats, there's no other predator that makes a full-time living on bats. And which is to say, there are a lot of things that take bats opportunistically, like certain raptors and owls, and snakes hanging down at roosts as they're flying out and so on. We'll take them occasionally, but it's all opportunistically. So there's no regular predators upon bats.
Huh.
They're not easy to catch.
No. And Well, I don't know. William is asking if the brain similarities with cetaceans. I don't know quite why, but
Oh, I don't know that I can comment on cetaceans. I'm not that familiar with their brain structure, but one of the things, like when I mentioned earlier how there was controversy on whether there were two origins for the bats, the Megachiropter and the Microchiropter. In the 80s, there was a controversy that with primates, one of the ways that we define whether something's a primate, like a little lemur or loris or something, is the visual organization of the visual cortex in the brain. And so in the early 80s, somebody looked at the Megachiroptera and realized, and look, well, their cortex is organized just like what we define as primates. And so there was a controversy whether the Megachiropters should be classified as primates and separate from the other echolocating Microchiroptera. So that just led to like, no, we got to define primates differently. And so, a lot of this has all gotten worked out, once we started doing molecular genetics and could track, similarities that way. Sure. But the brains of bats are very, highly sophisticated compared to, like, say, a rodent brain, and just the dentition. If you look at, like the dentition of a rodent, it's very simple. But bats, some of these bats have like 48 different teeth compared to humans 32.
They have that many teeth?
Yeah, different types of teeth.
Really?
Yeah. Yeah, from incisors to canines to premolars to molars. Yeah.
48 in the little body like that?
And yeah. Little micro teeth.
Holy Toledo. Wow. They don't want to run out, do they? And they don't have dentists, so it's like I've got.
I have captured bats still operating out in the wild with wizened, wrinkly faces and no longer any teeth. It's just they're worn all the way down to the gum, and they're still out there earning a living.
And they're just swallowing them.
Yeah, somehow they probably have like found their way to like go after the less hard prey items that are out there. They're resourceful. They find a way.
And in South Lake Tahoe, in the Tahoe region, for instance, with all those trees, I suppose the bats would very much use the bark of the trees and the crevices just like they would in Humboldt or
Yeah. And the larger the trunk and the more massive it is, the more it retains heat. And the ones that are out in the open and get more solar exposure are the ones that are typically preferred for roosts. But also at Tahoe, you got a lot of rock and rock faces.
Right, crevices, yes.
Yeah. And it's typically the southern facing rock, because then that gets the solar gain and warms up. And, on a very hot day, they just have to retreat deeper into the crevice. But when it's cooler, they'll come up closer to the surface where they can get the heat.
So do they actually move while they're sleeping, or is it?
Oh, not while they're sleeping. They route, like we wake up a little bit when we're sleeping and might roll over and move.
Right. Knock our spouse out of the bed.
Yeah.
Right. Edge over, right? Yeah. So they do the same thing. They sort of edge their way in and edge their way out, sort of.
Yeah. One of my grad students did work in a colony where they could, they were used to her being there. And so she could film them in infrared and, look at different behaviors. Like when the bat has to pee, if it's in the middle of a cluster, it'll actually like work its way out of the cluster and then turn over, hang by its thumbs, so then it could pee without getting it messing itself up or its friends in the cluster. It's
really
very fastidious.
Yeah. Very fastidious. Well, that's the first I've heard of that. I think that's very good indeed. I compliment them on great hygiene there.
Yeah.
It's remarkable what they do. Absolutely. Okay. Well, I think we should probably let's see. Okay. So other than I think we've covered the main questions, and I know I've got to let you go. I can't keep you forever as much as we'd like to, but Thank you so very much for joining us
My pleasure to join all of you.
and
Thank you
for
for the
solving
invite.
the technical problem that we had.
Yeah,
Thank
I'm
you.
sorry we didn't manage to work that out before, but
Yeah,
I'm glad we managed to get it.
right. Well, thank you again so very much.
You're very welcome.
All right. Take care.
Okay, thank you.
You're welcome.
Goodbye. Right, folks, I'm going to end the webinar here. And so we'll see you next time. All right. Take care.